much, Barbara. It's been absolutely wonderful working with you. All right, so we have some very popular pastimes that humanity engages in. Sports like this, and this cause tremendous amounts of joy and pain depending on which side of the field you are in. And one of the challenges has been that in this area, NFL has been leading the charge in denying anything bad ever happens in these sports. And there was a big focus on PBS a year ago, and there was a lot of media attention to this topic a while ago. So one of the challenges that happens in the traumatic brain injury is that it's very hard to diagnose, especially the mild cases, which account for roughly 75% of all traumatic brain injury cases. On the right is a chart from CDC that shows that not all of these are from sports, such as football. In fact, most of them are from causes that you and I would be quite easily susceptible to, such as falls, motor vehicle accidents, and so on. The collective costs estimated for these traumatic brain injury cases are roughly 76 billion annually. And the main problem in these cases is that they often go undiagnosed. So somebody hits the head, they go for an MRI. MRI doesn't reveal anything bad. They go home. And after a few weeks, they start to forget things. They start having cognitive impairments. By when any kind of interventional therapies are too late. So the question that we are all faced with is, are there modern kinds of imaging which will allow us to give an insight into what kind of visualizations can we have to diagnose the mild cases of traumatic brain injuries early enough so that interventional therapy can be helpful? So magnetic resonance imaging is one of these amazing therapies, one of these amazing imaging modalities in which you can have a peek into the functioning brain without any known side effects whatsoever. There are two kinds of imaging that are of interest to us right now here for the purpose of this talk. One is the diffusion tensor imaging, and the other is the diffusion kurtosis imaging. And these two kinds of imaging technologies give you volumes of data with up to 60 vectors per point. This is a crazy amount of data. It's very complicated. It's very hard to make any sense out of this. So let's first look at a much simpler case. Let's look at what is really the challenge in visualization of volumes with no vectors. This is a volume. It's a 40 by 40 by 40 volume that looks at the probability density of a two-body nucleon simulation in the 16 isotope of oxygen. Simple scalar field, and somebody comes and gives you this data and says, find for me what are the structures in this data. Where is the information in this data? So it turns out this is not so simple. So this is the next few slides from this package called Voreen that was published around 5 years ago in the IEEE Computer Graphics and Applications, just to show you how difficult the problem is. So here is a histogram. On the x-axis is the intensity ranging from 0 to 255. On the y-axis is number of voxels. Voxels are the volume elements of a volume. These are the grid points in a volume. The number of volume elements that have that particular scalar intensity. And so this is the kind of histogram you get. This curve over here is a simple map of opacity, meaning what do you want to make opaque in this volume? So if you have a simple curve like this, you see a simple blob like this. Not much to see. Suppose you take this curve over here and say, I'm going to increase the opacity, but only for the intensity values from here to here, and I'm going to increase it in this fashion. Put a small triangle there and say, this is how I will increase the opacity. Everything else is perfectly transparent. Only the small set of voxels whose scalar values are in this small range will be opaque. You see a sphere like this. Now, if you move this set of voxels that you're making opaque further to the right, from over here, you move them to over here, then you start to see this structure. And if you move it further out to the right, you see this torus. And if you collect all of them, change the amount of relative opacities so that they are different levels of translucency, color them differently, you can see all of them together. This is a hand-curated visualization. The question is, if you don't have the luxury of playing around with the small volume to your heart's content, what can you do? What kind of tools are out there to help you make sense out of this data? So what we did is we looked at this problem, and we found an automatic way to detect structures in these volumes. So I won't be able to go into details of that, but just to show you a simple video of what happens when you look at this. So here on the right is the interaction happening. And depending on what regions of this 2D histogram one clicks on, the program automatically does the segmentation and finds different clusters, different groups, different features of interest. So you can find skin, you can find skull, you can find sinuses and teeth. You can even find all the noise that is in a data set all clustered together. So that was a data set with just scalar values, basically one integer for every grid point. Now imagine what happens when you have one vector at every grid point. So here is a simple vector field. How does one visualize something like this? So the intuition or the inspiration for visualization of vector fields came to us, and by us I don't mean us here, but the broader visualization community, from looking at these astronomy images that track the motion of stars except without moving the telescope. So you keep the aperture, the shutter open for a long time, and you see this beautiful movement of stars. So the idea was that if you look at this image, there is no doubt in anybody's mind how the motion must have happened. You get a sense of the motion without looking at the vectors. So let's take this idea and apply it to vector fields. So let's take these vector fields and have a random initial starting point of intensities. And there's a process known as convolution, which basically says mix the two. And you get something like this. In this particular case, you are only doing this in a binary, star, no star kind of a method. But if you do it in a continuous case, you get these beautiful streamlines. This method was invented by Brian Cabral and Leith Leadham in 1993. Brian, at that time, was working for Los Alamos National Lab, and now he's working as the director of engineering for Facebook, doing very important problems of social connectivity. So that was one vector per point. Suppose you have 6 vectors per point. That defines a tensor, and it's often known as the diffusion tensor. Using those 6 vectors per point, you can define these ellipsoids. And in fact, this is what is used for visualization of these diffusion tensor imaging data sets. So you have, in the brain, every point has 6 vectors, and you combine those 6 vectors into these ellipsoids, fit a local ellipsoid, and you look at visualization of a cluster of these ellipsoids. The coloring of these ellipsoids is basically based on the orientation of the ellipsoid. Whether it's right to left is red, above up to below is blue, the third direction is green. So up to 6 vectors per point we can do. The problem is our collaborators in Baltimore are measuring at least 30 vectors per voxel. And this is known as the diffusion kurtosis imaging. What do you do with 30 vectors per pixel or per voxel? It gets really bad. So kurtosis is the 4th moment of statistical distribution. So let me just give you a very hand-wavy introduction to statistical moments. If mean were to be the center of a sphere, standard deviation would be given by the radii of an ellipsoid. Skewness would be measured by how an egg changes shape from above to below. And the kurtosis would be the peakedness of such a distribution. So we have settled the long-standing open problem of which came first. So this diffusion kurtosis imaging, the nice thing, why is it that these guys are so crazy about looking at 30, 40, 50, 60 vectors per voxel? So it turns out all these previous imaging modalities, the diffusion tensor imaging, for instance, was good only for the white matter. The white matter of the brain is the highways, the trunks, the tracts, the big fiber tracts. The gray matter has very subtle, very nuanced microstructure that is not visible if you only do diffusion tensor imaging. But if you do diffusion kurtosis imaging, measuring the movement of these water molecules under the presence of magnetic fields in 30 to 60 directions, you can start to see what is happening, what kind of structural changes are happening in the gray matter as well as the white matter. And gray matter is where a lot of the action also happens. So it's very important to study how the gray matter changes. So if you take this idea of fitting surfaces to these vectors, remember we had ellipsoids, these nice-looking ellipsoids for 6 vectors per point. So you take that idea and you fit these kinds of surfaces. In this case, they are from spherical harmonics, but you could use any surfaces. You end up with this clutter of all of these surfaces all over the place. And by the way, remember, this is only one slice of the volume of the brain. If you stack 100 of these slices, one behind the other, forget it. I mean, it's very hard to make sense out of this. So we were really stumped. So this happened to us around a year and a half ago. We were looking at this data and saying, how does one visualize 30, 40, 50 vectors per point? What do you do? So we decided to go back to the basics. What is really a visualization pipeline? It's really just 3 elements: geometry, camera, and lights. You move the camera around, you change the lights, you change the geometry, and your visualization emerges from the confluence of these 3 factors. So let's go to Hollywood. They usually define it the other way: lights, camera, action. So let's look at the lights. In all of these movies, this is a still from Ender's Game, they never make, these days especially, any shot with a single take. They take multiple such shots and then they combine them. And in the process of combining them, they make sure that the lighting is consistent. So in other words, this is a virtually rendered computer graphics generated object. This is a real actor. The lighting on this is from the left. The lighting on this would be from the left. You want to make sure that these are consistent, so there's no visual discrepancy that arises. And the question is this really important? So here is an example. So again, let's leave the issue of lighting aside. Let's go to geometry for a moment. In geometry, we have seen that local consistency is important, as in these case of Penrose stairs and ideas that were adapted from these by Escher's drawings on the ascending and descending. Local consistency is important. Globally, if things are not consistent, that's okay. It's not the end of the world. And so our human visual system cares more for local consistency than global. So now let's look at another example of this. So we talked about geometry. Let's talk about cameras. So here is a modified camera. What it's doing is it's a multi-perspective camera, which as you move around the head, it's taking one row of pixels in the middle and then compositing them. There are examples of this that are being used to do multi-perspective street views. So at a single view, you see all the storefront windows. Again, things are locally consistent with what we would expect. Globally, they are not. And it's a little bit bothersome, but not too much. We can still make sense of what is it we are seeing. And there are artists like Rob Gonsalves who combine the local consistency of geometry and camera with global inconsistency. And the results are, locally, they're fine, they're appealing, in fact, in some ways. So the question for us is, if the geometry and camera can be locally consistent and globally inconsistent, what do we learn about this, about the nature of lighting and our perception of lighting? So here are examples where Photoshop has been used to locally change the consistency of lighting and see if you can spot these inconsistencies. And it takes a while. It doesn't obviously jump out at us. So for instance, I'm sure some of you have seen this at this point, the arches of the Taj Mahal are in this side, shaded over here, but they're shaded on the opposite direction over here. You can't have a mountain lighted from the left and the right at the same time, and similarly, there are similar inconsistencies here. It's very hard. So can we use this to enhance the perception of these really intricate geometry, really intricate data? So there was this study done by Ostrovsky, Sinha, and Kavanaugh in 2006, published in this journal Perception, where what they did is they oriented these cubes randomly, and they asked people, do you see which cube is lit inconsistently with respect to the others? And what they found is, on an average, users took 8 seconds to answer this and were then wrong 30% of the time. So in this particular case, this is the one that is lit inconsistently with respect to others. So good. Lighting inconsistency should not bother us at this point. In fact, artists have been using it for a number of years. So this is the painting of George Washington crossing the Delaware. This was way back when Delaware River used to get frozen a lot. And everybody is lit from the left, except for this guy who's being lit from the right. And there are lots of people who theorize, who hypothesize about these things. Why is this guy being lit from the right? And the general consensus amongst art historians is that this is to give, at a subconscious level, a notion of brilliance an effulgence to George Washington, who's radiating this light outwards to those around him. Same thing happens when you look at this classic painting of the music lesson by Jan Vermeer. At first, you ask people, what is inconsistent? And whenever I've asked this question in my class, most people do not think of lighting. People who look at it very carefully would say, well, the girl in the mirror is inconsistent with respect to the girl here. She is looking to the left, she is looking straight down. In fact, X-ray radiographic analysis of this painting does show that initially, when Vermeer had painted the original version of the painting, this girl was looking to the right as well. And then he erased and made her look straight, but didn't bother changing the reflection. But that's not what we are talking about right now. We are talking, that's a geometric inconsistency. We're talking about lighting inconsistency. And the lighting inconsistency here is, this shows the sun is up there casting a shadow here, the sun is up there casting a shadow here. These are consistent, but then what happened to the shadow over here? There's no light, and there's no shadow. Something is a bit strange. The general interpretation of this is that this was done to avoid distracting these shadows from here that would have been cast here and over here that would have been distracting, and instead allow the focus of the attention to be on the girl who's in the middle, and nobody's looking at, and everybody's sort of looking at her from the different directions. So now let's go to scientific illustration. On the left is a photo of a skull being lit from the left. On the right is an illustration, a hand-drawn illustration. This comes to us from this book by Hodges in 1989, The Guild Handbook of Scientific Illustration. And notice that there isn't one light source. There are multiple light sources. There is one light source from here, and there are lots of these local light sources that are being used to light the skull from different directions. Otherwise, the skull would have been lit like this. And this is used to highlight and emphasize different aspects of the structure of the geometry. And it doesn't bother us. It is so strange. You would expect that after millennia of evolution, on a planet that has one sun, you would be distracted if you have multiple local light sources. But I guess there were lots of surfaces of reflection in the world around us, so we never really got bothered by it. So let's use this. So the first thing we did is, let's say, instead of one light, let's have lots of lights around 3D objects, and let's light them and see if you can emphasize the subtle structures of the geometry. So here's a manuscript. It's a 15th century manuscript. This has been scanned to about a 5 micron accuracy. So this 5 micron depth accuracy is fine enough to pick out the differences in the level of the ink in this micron. If you have one uniform light source, that's how the manuscript looks like. If you have 4 uniform light sources, that's how it looks like. If you increase the number of light sources, it just gets worse and worse from here on out. But if you have 4 lights that are locally changing, in other words, you look at one region of this manuscript and you say, this region of the manuscript, because of this algorithm I have, will pick a light source from this direction. An object a region of this manuscript right next to it will get a light source direction from here, and you find local lighting. Only 4 light directions, except they are being very carefully positioned for every local region of the surface. You get both the low frequency details and then the high frequency details. And I thought this was a really cool idea. So a few months after this, I met Tony DeRose from Pixar, and I said, hey, we have this really cool idea. We light things locally. And he said, what's new? We do it all the time in Pixar. So except we don't publish it. So this is a really good idea. So now let's see if we can use it for our brain imaging. But there's one more thing I need to talk about before I get to that. And that is this whole idea of spherical harmonic lighting. So spherical harmonics are these basis functions on a sphere. You have probably studied it when you studied chemistry. The s orbitals, the p orbitals, the sp2 orbitals. In 2000, two groups of people completely working independently with each other, with respect to each other, found that spherical harmonic lighting can be used to define how light reflects off of people's faces. One was Basri and Jacobs. This guy, Jacobs, by the way, is now with us at the University of Maryland. At that time, he was working for NEC, and now he's been working on some amazing applications on leaf snap. So you take a photo of a leaf, and it'll tell you what tree it came from. So go to leafsnap.com, and that's David Jacobs. The other pair, Ravi Ramamurthy and Pat Hanrahan, are from Stanford. So instead of lighting, the basic idea here is very simple. Instead of lighting with a point light source, such as over here, light in terms of spherical harmonic lights. So here is the exact sphere that you're trying to replicate. These ones got squished, but if you only use the top, the first term in the spherical harmonic lighting, light it equally from all directions, you get this. It's the root mean square error, in this case is 25%. If you light it using the first and the second bands of the spherical harmonic lights, meaning uniform from all sides, differentially from above and below, front and back, side to side, you get this, which is root mean square error 8%. And then if you use all three bands, 9 terms, you get really good. This only works for diffuse lighting. So if you have Lambertian surfaces, diffuse lighting, if you have shiny specular surfaces, it doesn't work that well. But most of us, most of the time, unless we have bald heads, are perfectly fine with diffuse lighting. So here is how the spherical harmonic looks like. So if you take the grand challenge application of visualizing an armadillo and light it with the spherical harmonic lighting, green from above, blue from below, a point like this will be lit green, and a point over here will be lit blue. And you can even look at the decomposition of lighting using spherical harmonics on this armadillo along those first 3 bands. So it turns out the spherical harmonic lighting in the last decade or so has really taken off. You can use it to light very sophisticated lighting algorithms in graphics with the very nice environmental conditions very, very fast. So almost any game you or your teenager plays is likely to have spherical harmonic lighting implemented in it. Here are 2 screenshots from Xbox's Fable Legends. On the left is a scene rendered without any spherical harmonic lighting. On the right is the scene rendered with the spherical harmonic lighting turned on. So you would notice, see if you can see how the subtle structure of the grass and foliage over here comes up compared to over here. And again, it's really good at bringing out these features. So let's now, we have looked at inconsistent lighting, we have looked at spherical harmonic lighting. Now let's apply them all to the 60 vectors per voxel problem. So this is what we did. You get the data from this diffusion MRI, you break it up into these spatioangular fields calculations where you're computing a kurtosis tensor for every voxel, and now you use lighting which changes locally depending on the characteristics of the underlying geometry to light these objects. So here on the top are the kurtosis tensors expressed in the spherical harmonic basis functions. This, by the way, is another way to express spherical harmonics. I mean, basically, in these, if we were to take the radius of the sphere to be the intensity, you will get things like structures like this. Just another way of looking at spherical harmonics. So here are the kurtosis tensor spherical harmonics basics. Here are the lighting functions. And to light any one of them with any of them, you just do a simple dot product, just a small number of multiplications and additions, things that the graphics hardware can do really fast, really well. And here are the color tables that you get out of it. So if you have, now let's take one slice of the brain with the kurtosis values expressed all over these slices. If you use the spherical distribution, the brain looks like this. If you use a directional distribution, the brain looks like this. If you use a radial one, it looks like this. If you use 2 lights, it looks like this. And if you use 3 lights, it looks like this. Now, one thing I mentioned, and oh, by the way, here is a directional spherical harmonic lighting for a traumatic brain injury patient, and you start to see the traumatic brain injury really well. On the right is this video showing how fast this whole process is. So instead of going to streamlines, I mean, imagine having 60 streamlines all going across each other, you can get the same idea by moving the light source around the kurtosis tensors that are sitting on one slice. So here is the lighting, the 3 lights, red, green, blue. They were being rotated, and you saw that. Now let's look at a slightly different version of things. So what we just did in the last set was simply do the lighting by the 3 lights. What we are doing here is taking the light response and mapping it into color and opacity, not just color. So things which are being lit more are becoming more opaque. Things which are being lit less are becoming more transparent. And now, instead of looking at one slice, you can look at the whole volume. So this looked really good to us. But the question was, is this really useful to anybody? Oh, before I go to the usefulness part, one thing I forgot is, remember we were talking about the inconsistent lighting? So the light orientation, if you do a global light orientation, everybody's aligned exactly the same way, you get this. If you change the light locally, depending on the local kurtosis tensor, from point to point, so these are lights that are changing locally, and then you map the resulting light response to the opacity of the surface, you get much more detail. So we took this method to our collaborators in Baltimore, and they gave us these amazing data sets. They said, see if they work on these data sets. So on the left is what I would say is the current state of the art. These radiologists are looking at mean diffusion, fractional anisotropy, and the principal diffusion directions. On the right are our 2 visualizations. And it's so much easier, I would claim, to look at the traumatic brain injury using these kurtosis values here and here, and even more clearer if you look at them in the color spectrum. These are simple color segmentation based on kurtosis values. So the second case study we did was on a mild brain injury where the MRI was taken 10 days and 6 months after the injury. And the concussive symptom was that there was a decline in cognitive function. And we wanted to see if it would be reflected, if it would appear in our visualization. So on the left is the brain function after 10 days, and on the right is the brain function after 6 months. And you could start to see how the brain in these kurtosis tensor visualization has deteriorated. The next case study was the opposite of this, where there was a frontal lobe damage, and there was impressive recovery after 1 month in the patient. However, the conventional MRI was unable to show this. So on the left is this fractional anisotropy from the diffusion tensor imaging. 8 days and 1 month after, they both show a substantial loss of matter here. So what we are doing is really we are using our lighting technique to give the other technique the best possible advantage in this case. And here is the kurtosis data showing recovery in the brain tissue over here. The next example we tried was the case of tumor. And in this case, again, mean diffusion, fractional anisotropy, and the principal diffusion direction do not show the presence of a tumor as well as our visualizations can show. So clearly, it does seem like there is some value. And so at this point, we asked our radiology collaborators to come speak to you about what they see.
Sujal's work is extremely important for us because it allows us to visualize these microscopic or microstructural changes in the gray and white matter at the same time, and also be very close to the basic science where you're doing microscopy. So this opens up a window for us to visualize microscopic processes in the brain that we were otherwise unable to do in the past.
So as compared to diffusion tensor imaging, diffusion kurtosis imaging deals with a much more complex tensor structure, the kurtosis tensor. Currently, there's not yet a very good way of visualizing that. Sujal's work is a breakthrough that finally provide a way to visualize the kurtosis tensor in a very useful way and then very intuitive manner that will help the radiologist to find abnormal areas and then to understand what's really going on in the tissue microstructure.
So Sujal is the PhD student who led this entire visualization study and who you saw in the video. So the basic contributions here have been to use the spherical harmonic lighting to depict spatio-angular fields that are reasonably dense. And the interesting part is that in the past, people have focused a lot more on visualization using changes in the camera or the geometry, but one of the things which is interesting here is that lighting can help draw out these anomalies much better. In this particular case, especially when the data complexity becomes very high. And one of the things we are working on now is to establish this kind of a visualization with further studies as a useful clinical diagnostic tool for looking at and interpreting these large-scale diffusion kurtosis imaging data sets. So one of the best things that is happening these days in the life sciences and in biomedicine is, on one hand, we are being flooded with massive amounts of data in the volume, in the variety, in the complexity. At the same time, we are also very fortunate to have an amazing set of advances in the technologies that will help make sense out of this large amounts of data that is coming our way. So here is a small gyroscope. A smaller version of this has been integrated into your iPhone and an accelerometer has been added to it. These are tangible interfaces that, when combined with visualization, will be able to help us make sense out of these massive amounts of data that are coming our way. One of the challenges has always been, what does a person see? What does a person understand when they look at a visual image? We have used eye tracking in the past, but now there are these amazing advances that have happened where you can look at consumer-level EEG data. You can look at consumer-level FNIRS data, the functional near-infrared spectroscopy, to try and make sense of the level of comprehensibility of the visualizations that are being presented. For the first time, we are able to get a quantitative feedback of what somebody is seeing and how much they're comprehending, and this can be used as a way to control the rate of information that is flowing to the user. If you don't want too much information that overwhelms a person, you don't want too little information that people find boring. So this is leading us towards the next generation of visual computing tools and technologies that will be critical to help make sense out of the large amounts of data that are being generated. And towards this end, one of the things we are working on right now is building an interactive display environment where we'll be using projectors, robots, and the sensors to sense motion of the eyes, of the hands, of the head, to be able to interact with ever-increasing amounts of data. This is a project that that was seed funded by the college, as well as we recently also got funding for this from the National Science Foundation under the Major Research Instrumentation Award. So in some ways, these are very exciting times, and I look forward to being with you in this very exciting journey. Now, a lot of what I talked about today was not my work. It was work sponsored by these folks and done by a lot of very nice people who have been spending their time accompanying me on this journey. So I look forward to working more with them and with all of you. Thanks.
So I'm curious, what kind of curriculum and courses and so forth that computer science is teaching these days to prepare students for this type of research?
That's a really good question. So right now, there are lots of programs where we are working on topics such as highly parallel processing, machine learning. At the same time, at some level, we need to move away from the conventional style where you are teaching people to become computer scientists, or you're teaching people to be biologists, or teaching people to be artists. And I think we need to go towards this newer style of a campus, a newer paradigm of a campus where one can create opportunities for students to blend their interests and abilities that span multiple traditional disciplines. And there are discussions that are going on right now to start majors and minors in computational biology, in digital media design, major and a minor. And so hopefully in the next few years, we will start to see more of these cross-disciplinary educational opportunities. In the past, we have been successful in building bridges across communities in research, but not as much in education. And I think moving more towards interdisciplinary, cross-disciplinary, and transdisciplinary vocations would be really where we should be headed. And I think this campus is really amazing in how it's adapting to these newer challenges. Cybersecurity is yet another field where we're bringing together people from lots of different areas. There's an amazing ACES program for undergraduates, which brings together students from different backgrounds. So I think these things are beginning. And I think Maryland would probably be one of the leaders in this. Thanks. Hi.
So I have a question about when you change the lighting, are you looking at the transition from like the gray matter to the white matter, the CSF, or are you using like an internal lighting? Are you using T1 and T2 weighted images and looking at the gradients? How do you make lighting and changes?
That's a really good question. So what we are doing is we are orienting the light by the local diffusion directions. So of these 60 vectors, we fit an ellipsoid, find the 3 principal directions, and orient the light using the principal directions. But what they're lighting is really the full kurtosis tensor.
So in terms of, say, taking the traumatic brain injury as a case, we've generated visuals for which help us as humans to see this. Any progress on making, say, an algorithmic interpretation of it, or something that doesn't necessarily take a human looking at it to determine what's normal and what's damaged? Or
That's a really challenging problem. So here's a fundamental difference between where you are headed and where I'm headed. The difference is I look at computers as assistants to experts. So my view of computer science is that it's a tool for intelligence amplification of the human. Not to have the computer do the diagnosis, but to help the humans do the diagnosis. And I think that's a difference. So in that intelligence amplification view of the world, the basic hypothesis, I guess, is that a human with a computer working together will always be more powerful than either alone.
fair enough.
Thanks. But that's a really good question.
One of the faiths of big data, the most important aspect of it is that almost all of it's irrelevant. And so, well, you were picking out a signal in the brain, and if you're just given this huge amount of stuff, or we were hearing it bacteria in the gut and so on, how do you find the signal? So you keep saying that you have this 60-dimensional vector, but you don't want a 60-dimensional vector. You are using some little aspect of it, right?
Yeah, absolutely. In fact, so I think what you said is really good. If, God forbid, we had to look at 60-dimensional vectors, we would be in big trouble. And I am so grateful that nature has these lower dimensional subspaces where all the useful information resides that make our life so much simpler. Because you're right, if things were to be really high dimensional, then it's very hard. It's very hard. Now, the other thing which I feel like I must add is one of the, and I didn't talk much about this today, but one of the cornerstones of a successful visualization is finding what is salient in the data and emphasizing that visually. Because unfortunately for us humans, we only get, attention is a zero-sum game. And you can't pay attention to everything. And so, but that's very well taken, yeah. All right, well, thank you very much.